Thank you for joining us today. I am Dina Baptiste, a member of the GIN North America Steering Group and Director of Cancer Screening Guideline Development at the American Cancer Society. Today, I will be moderating the webinar on bringing evidence and guidelines into the digital age. As we have new participants today, please allow me to take a moment to share some background information with you. GIN North America is a regional community of a larger GIN organization and include members from Canada, the US, and Mexico. This webinar series is one of the ways we facilitate sharing and collaboration in our regional community, and we encourage you to visit the GIN North America website regularly for information on upcoming events in our regional community. A few housekeeping items before we get started. This webinar is being recorded, and the slides and recording will be available on the GIN website in about a week or so. Also, you may access all previous webinars on the GIN North America, on the Guidelines International Network website. The link is on the slide presently showing. Please note that all participants were muted upon entry to the meeting today to facilitate a smooth recording. We have allotted time for questions at the end of the presentations. So throughout the presentations, we ask that you please submit your questions in the chat section. I will convey as many questions as time allows to our speakers at the end of the session. Now, let's go to today's talk. For our GIN North America webinar series, in addition to featuring ongoing and completed research, we also work to feature innovative tools and resources available to guideline developers. Today's webinar is an update on freely available tools and resources that can assist in adapting evidence and guidelines for the digital age. Our speakers will describe the context of using these resources for integrating the computable and narrative aspects of guidelines and evidence. Our first speaker today will be Dr. Brian Alper. He is board certified in family medicine and clinical informatics. In 1995, Dr. Alper founded Diomed, an evidence-based clinical reference with the mission to provide the most useful information to healthcare professionals at point of care. More recently, he started scalable efforts to define the standard for computable expression of evidence and statistics with a project to extend fast healthcare interoperability resources for evidence-based medicine knowledge assets. In 2020, to scale these efforts for immediate needs, he started the COVID-19 Knowledge Accelerator, changed his mission to enable standard-based machine-interpretable expression of knowledge, especially related to healthcare and scientific evidence, and founded his new venture, Computable Publishing, to fulfill this mission. Our next speaker today will be Dr. Ian Saldana, an assistant professor and evidence-based health researcher at the Brown University Center for Evidence Synthesis, where he serves as assistant director of the AHRQ designated Brown University Evidence-Based Practice Center, and also as director and principal investigator of the AHRQ funded Systematic Review Data Repository Project. His research focuses on the use of outcomes in comparative effectiveness research, methods, and technology for research synthesis, and methods for research prioritization. Also today we have Ms. Maria Michaels, who serves as a public health advisor at the Centers for Disease Control and Prevention, where her work involves bringing together health, information, technology, healthcare, and research perspectives. She has served as technical lead and program manager for high-tech clinical quality measure policy and operations at the Centers for Medicare and Medicaid Services, and also as program manager at the National Cancer Institute Cancer Human Biobank. Ms. Michaels is also a member of the GIN North America Steering Group. Our final speaker for today will be Dr. Edwin Lomaton. He is board certified in pediatrics and clinical informatics and currently serves as Chief of Clinical Informatics for the Division of Digital Healthcare Research in AHRQ Center for Evidence and Practice Improvement. Also, he currently leads AHRQ's Clinical Decision Support Initiative, which aims to advance evidence into practice through clinical decision support and to make this more shareable, standards-based, and publicly available. Now I will turn things over to our speakers. Dr. Alper.
Okay, thank you. So I'm excited today to share with you a number of thoughts on bringing evidence and guidelines into the digital age. Starting with some introductions and disclosures. As mentioned in the opening, I've started a new venture, Computable Publishing, which is a company designed for the purpose to augment digital publishing with machine-interpretable forms and offer anybody looking to use these materials tangible ways to get that done. The tools developed here will be made freely available for COVID-related knowledge, and the efforts of COVID Knowledge Accelerator and EBM on FHIR, which I will discuss in more detail in this session. These projects are completely open, transparent, no cost, collaborative, active efforts. So the AHRQ has a large effort advancing or achieving evidence-based care transformation support, or the ACTS initiative. And there's a grand vision across the entire knowledge ecosystem to smoothly move through data generation, evidence generation from the data, guidance generation from that evidence, decision support, make that computable, action in terms of better healthcare coming from all of this, and the result of that leading to data generation and going all the way around the cycle again for this learning health system model. And this should all be advanced to use the tools of the digital age. So everything you hear through the rest of these presentations could be seen as components or pieces of this grand knowledge ecosystem view. But where are we with it today in our healthcare knowledge ecosystem? We have more data than we have ever had before, and we'd like to harness that and analyze that and transform that into evidence. But at the moment, getting from this data to that evidence is not easy. It's as if we have to individually deconstruct the data sets for each data point, figure out what it means in the way we want to analyze it for evidence, and then reproduce it in this evidence form. We finally do that, and it's a lot of work, to create the evidence reports and create the systematic reviews of the evidence reports. And after all that heavy work is done, wouldn't it be nice if we could just say, now, what should we do? What is the guidance? However, pardon me, the GoToWebinar viewer got in the way of my screen. Are you seeing the data evidence guidance action, the full series? Okay, I will keep going and just do not have the screen in front of me as I look sideways to describe it. What I was going to talk through is each one of these steps, getting from data to evidence, getting from evidence to guidance, and getting from guidance to action, unfortunately, is today an intense effort of finding that item, finding the specific detail we need in that item, deconstructing it, putting it together for the next thing, and reproducing it. And if we look at all of that, we end up with spinning our wheels. Doing this effort does not move the needle, get us to the next part, and the different parts are not well coordinated. What we would prefer, the desired knowledge ecosystem, is each of these major efforts, the effort to get all the data out of the system, the effort to analyze all of that data and synthesize it into evidence, the effort to take all that evidence and other inputs and create the guidance, and the effort to take all that guidance and make things happen, the action of healthcare. We want that to be interchangeable, each smoothly interoperating with the next part, and this is the ideal or desired healthcare knowledge ecosystem. Another view with the images adapted from the Magic Foundation, another group involved with many of these groups that you'll be hearing about throughout this session, working together to envision this digital and trustworthy evidence ecosystem. And you see, we produce the evidence, synthesize it, produce the guidance, get it into healthcare. What are we creating at each of these steps? For the focus of this session, I'm going to focus on producing evidence or research studies, synthesizing it or producing systematic reviews, and producing the guidance, which may look like guidelines, decision aids, or clinical decision support. How do we make it better, faster, easier, more accurate to make everyone's life easier throughout this ecosystem? Key is to look at the data. What data is being transferred at each of these parts? What data do we need to take the results of research studies and create systematic reviews? What data do we need to take the systematic reviews and inform and create guidance? What data do we need from that guidance to disseminate it, to implement it, and make this happen? If we look at each of those steps of data transfer, we can use standards, technical standards, to approach the data in the same way so that we don't have to do that heavy work of deconstruction and reconstruction every time we want to transfer the data. Applying standards to a domain can be broken into 4 parts: the schema, or common format for how data elements are expressed, the terminology, the actual terms, definitions, codes, code systems, value sets used, the tooling, whether it's user interfaces, what you recognize visually as the tool you use, or APIs, the tools for how computers work with each other, and many, many variations for specific content, specific people, specific groups for contextualizing, applying all of this. That's what we need to do to make it easy. Health Level 7 International, HL7, is a standards development organization, an organization set up specifically to produce standards in this way to help at a global level, how it all fits together. ANCI is an association that even accredits standards organizations, standards development organizations for how to do this. HL7 is focused on healthcare standards and has had very wide global contribution and use, and is well known in the healthcare space for supporting data exchange. If you have not heard of any of the other HL7 efforts, you may have heard of FHIR, FHIR. FHIR is exciting in that it is overcoming the challenge of speaking between electronic health record systems in a way that has never been achieved before, and it's really happening now. FHIR stands for fast. We want to make it fast to work in real time. Healthcare as the focus. Interoperability. We want everything to work with each other seamlessly. And resources. The atomic unit that is shared in data exchange is called a resource in FHIR and has standards for how it is expressed to make this work. So FHIR is now completely open and widely used and effective for bringing an interoperability to healthcare. What we want is to apply standards to the evidence domain. All the work we do to produce the evidence and research results, to produce the systematic reviews and understand how to interpret all the evidence we have, to produce the guidelines and take this evidence and transform it into making recommendations. That world that we could call evidence-based medicine is related to but different from the world of healthcare. The EBM world has wanted interoperability for a long time, and I've been involved in multiple ways saying, how do we make that happen? In 2017, at a GIN meeting, the tech group I was charged with helped us take some first steps in interoperability for the EBM community. And in 2018, I learned enough about FHIR and how it is solving the interoperability problems for healthcare and said, why can't we use that instead of having to recreate even the method of how do we tackle interoperability? So started a project in 2018 to extend FHIR standards to evidence-based medicine knowledge assets. EBM on FHIR is the short or nickname of this project, and it is to extend FHIR resources to express research and findings, evidence, and recommendations. In 2020, with COVID giving obvious needs in so many ways and so many large groups working together like never before to advance knowledge related to COVID, there were many requests to apply the EBM on FHIR developments to COVID-related knowledge. And we ended up creating COVID Knowledge Accelerator, an open virtual organization that now has 10 active working groups, meeting 11 times per week and doing many things, as I'll show you through this presentation, to advance standards for expressing evidence and recommendations in FHIR standard format. So here is a view of the schema or model for a evidence resource. And you can express evidence by expressing each of these concepts in a defined way. And I've highlighted statistic as one component of the evidence to be expressed. The statistic data type has many elements within it. And this is what it may look like to see in the computable code of a specific statistic. You'll see here a description in human readable form, the age-adjusted rate ratio and some numbers, and then the actual elements. It is an incidence rate ratio and the quantity is 1.15 and there's a certain number of events and the attribute estimate for the confidence interval. If we take a look at that coding just for the concept of what is this statistic and this specific code to say it is an incidence rate ratio, that leads to another part of the standard development. We need the terminologies in addition to the way we package the data for electronic expression. So the COVID Knowledge Accelerator is not only developing many resources like evidence, evidence variable, evidence report, and citation, it is also developing terminologies. And we have multiple code systems to identify those terms that we need to uniquely express. Here is an example view of the statistic type code system that we are developing, and you'll see incidence rate ratio towards the bottom of that list. We have 89 concepts right now for 89 different statistic types. We are creating code systems for study design concepts. Is this randomized? Is this parallel cohort or crossover? Is it cluster assignment and many other study design concepts? Statistic types, as you've seen, statistic model that needs to be codified as well, and a risk of bias code system that at the latest count is up to 225 different discrete concepts for expressing a risk of bias. You can use that tiny URL, invite to EWG if you want to join an expert working group and be part of creating any of these code systems. We then have tooling, and I'll show you some screenshots in a moment of Clinical Trial Results Reporter. That is a tool that lets you enter the data easy for human form, and it will convert it to these FHIR resources, and you do not need to know any computable programming to create the computable expression of the evidence. You could participate as a user, simply using the tool and giving feedback to improve it, or as a researcher, and we can develop feasibility and usability studies to advance this tool development. So here's an example from the first page, Clinical Trial Results Reporter, acknowledge the Lister Hill Center of National Library of Medicine, some partial funding and support as we were adapting this for clinicaltrials.gov results reporting demonstration. So the first page, you may enter some data. What is the population for this study? What is the intervention? What is the comparator? Here's an example we used for the clinicaltrials.gov demonstration of the adaptive COVID treatment trial with some simple descriptions of the population and the remdesivir and placebo with the dosing in the description. On the next screen, you can enter baseline measures, and we can set whether it's a dichotomous measure or a continuous measure, what type of statistic we're reporting for continuous measures, and then places to enter all the data. Here is an example with data filled in for the baseline age as a continuous measure and categorical age between 18 and 65 and age more than 65. And then you see some other dichotomous or categorical measures on this page. Finally, we can enter some outcome measures. And in addition to things you saw for baseline measures, you can now enter the time from the start of study to specify the time the outcome was measured. And then here's an example with outcome measures entered, and then risk differences or mean differences calculated from the available data in this example. And then you can see a report that puts all of this data together. And this is a human readable summary of the clinical trial results that were reported. What is special about this tool is in the upper right where you see convert to FHIR, it will convert this data set to the many evidence resources, evidence variable resources, and evidence report resource to package all of this in a computable standard structure for sharing this data. Finally, an example of specific content and people that we are doing right now as a demonstration project for the COVID Knowledge Accelerator is a systematic meta review of steroid therapy for COVID-19. So we are seeking studies where the study method is a systematic review of randomized trial, and the PICO is for the population symptomatic COVID-19, the intervention steroid therapy, comparator no steroids, and we're seeking any clinical outcomes, and we've defined a list of specific outcomes that we care about. So the goals of this systematic meta review are: one, determine the consistency of findings and judgments across systematic reviews for this important therapy for COVID. If there is inconsistency, we are not going to make the call over which judgment is correct, but we will determine the most current accurate findings from this evidence, and just reporting the consistency of current systematic reviews for this will be helpful. But goal number 3 is for all of this data, we want to document the findings in computable resources. We'll make many FHIR evidence resources to express all of these findings. And what that means is efficiency in updating. If we want to change anything, like making a living systematic review, it will be efficient to make adjustments. If we want to share this data, we'll have an efficient way to share the data. And as we go through this in a little more detail, these things may be useful to you if you're involved in joining this project to create the systematic meta review. And also, if you want to be notified as results are created from this systematic review, because you can use the results that we create. So we have 9 databases, 10 if you count Prospero as a secondary way of finding the systematic review published results, that are out there. And we are going to publish the protocol. We've created the search strategies. We're still filling out the rest of the protocol for the systematic meta review. And we will be mapping the search results to citation resources for each of the articles found, and deduplicating the citations, and having systems to manage the screening or inclusion and exclusion decisions for the systematic meta review. So here is a view of the initial searches of the 9 different search strategies. Found 1,254 articles, but after deduplication, which happened automatically with Pico Portal, we have 656 unique articles to consider for screening for this systematic meta review. This is a quick screenshot of some highlighting we get to help us decide what is the Pico elements. Is it a systematic review to meet our inclusion criteria? Next, we need to do data extraction. And the 9 elements here are the 9 things we will specifically extract from each systematic review that is included. The specification of the population. So we could separate, for example, inpatients from outpatients if there's data for both groups, or those needing oxygen therapy and those not needing oxygen therapy. The actual specifications will depend on the data that we find. Specifications of the exposures or interventions and comparators. And we may find that different dosing or different drugs have different results. Specifications of each of the outcomes. So those first 3 concepts are definitional descriptions to make precise data capture. Then for the statistics, we want the value of the statistic itself. Is it a relative risk, and what is that point estimate? The sample size behind it, and then the any statistic attributes or model, the confidence interval, the p-value, model adjustments for getting to that. So we'll be doing a lot of coded data to really understand the statistical results of these systematic reviews. And then to understand the certainty of the evidence, we'll have to code what is the certainty rating system used. Maybe that review used the grade rating system, maybe they use another system. And then what is the certainty rating by that systematic review author using the system that they use to express it. And finally, we will do our own risk of bias assessment of that systematic review using the Robust tool and have to express our risk of bias findings. So all of these 9 things that we will be doing data extraction, we will want to create data entry support. So I just showed you the clinical trial results reporter. You can imagine a systematic review results reporter where we need to report the results of the systematic reviews. And there are differences in some cases than reporting the results of clinical trials. So we'll need to create tools to facilitate this effort. Then we will create the content in coded structured form, specifically using FHIR group resource for populations, evidence variable resources for the exposures and outcomes, and evidence resources for the statistics and certainty, and then evidence report resources for how it all wraps together. As we create those things, we will then be in a good position to develop systems. So when the technological thing changes by some criteria, you could be notified instantly that this evidence changed, evidence related to this group changed, or whatever that may be. And hopefully you can imagine how these things may give you some individually specific support if you are looking for this type of data Maybe you have a guideline related to steroid treatment for COVID, for this actual example. So
Dr. Alper, 2 minutes.
Yeah. Okay, this is, I believe, actually my last slide. So very good for the timing. So the easiest way to participate in the COVID Knowledge Accelerator is to come to one of our open project meetings. You can go to tinyurl.com/COKA2020 to find the schedule of meetings and the link to join them. And you can also email me, my email, balper@computablepublishing.com, if you would like to be put on the distribution list. And I send an email every day with an update on whatever we did today across the COVID Knowledge Accelerator. You can see with these many different groups, there are 4 of them that say something code system development, statistic model, study design, statistic type, and risk of bias. Those 4 groups are developing specific terminologies for those 4 code systems. The systematic meta review is obviously working on the steroid for COVID systematic meta review project. You see 2 tools development groups, evidence evaluation and reporting, which most recently is focused on the clinical trial results reporter, and content citation and classification, which is what resulted in that PICO portal view that I showed you for how we handle the citations for the systematic meta review. There's a communications group where we seek to help anyone across the COVID Knowledge Accelerator for making scholarly publications or presentations like this one, or other awareness of this overall effort. And there is a knowledge ecosystem liaison, where we often connect with the ACTS project, and could be any of the other projects that relate to this area. So we could shift now. I believe Ed is next in the series.
No, I think it's Ian.
Oh, okay. My apologies. So thank you for that.
All right. Good afternoon, everybody, or good morning or evening, depending on where you are. So once again, so my name is Ian Saldana, and I'm at the Brown University Evidence-Based Practice Center. And what I was tasked to do in this presentation is provide you a little bit of a deep dive into the Systematic Review Data Repository, which is one of the platforms that systematic reviewers use to conduct systematic reviews. Specifically, what I'll be speaking about is some of the details about the platform itself in order to help us think through what comes out and how it feeds into the rest of the ecosystem that Dr. Alper just spoke about. So before I go further, I wanted to acknowledge funding from AHRQ that has funded SRDR, or the Systematic Review Data Repository, right from the beginning when it was launched in 2012. And I'm the lead of the project right now. And that's the contract number. But other than that, I don't have relevant conflicts of interest. But I'm speaking on behalf of the systematic review team on from Brown, and not on behalf of AHRQ. I always like to talk about software first by the people who work on the software. So this is the team that we have here at the Brown Evidence-Based Practice Center. It's a team of a mix of systematic review experts, some clinician epidemiologists, some and programmers that we have here, and research associate staff as well. So we are the team that works on the tool and supports its use. What I'd like to talk about is why the tool exists, a little bit of an overview and a quick demo through the tool using some screenshots, and then talk about one instance of the sort of thing that Dr. Alper was talking about, how we are using data coming out of SRDR Plus to hand it off to the next piece of the ecosystem. I'll use the outcome definition viewer as an example, and I'll end with some closing thoughts. So why does this tool exist? So back now more than a decade ago, there was early recognition by quite a few people in the field, led by especially Dr. Joseph Lau, who I'll talk about in the next slide. There was a strong recognition that systematic reviewers must be transparent. We need to collaborate widely. These are big projects. There's a lot of data that goes into it, comes out of systematic reviews, and spreadsheets or paper-based systems, etc., get unmanageable in this process really fast. This is the type of effort that takes a lot of costs and time. And in an ideal world, the data that are collected and used in systematic reviews should be reused, archived, and hopefully shared with other users and other organizations. So the vision was to create, and that was Dr. Joseph Lau, the original, sort of the original architect or the original founder of SRDR, with the funding from AHRQ. He's now retired, but his vision continues in the work that we do to maintain SRDR as a free open source platform that serves two main purposes: the data management process during the systematic review, that's screening and data extraction, but also this aspect of reusing data down the line, perhaps by the same team or other teams, archiving data and sharing it more openly. So with that sort of vision, what we've viewed at SRDR over the years is as a community resource for the community of scientists around the world, as well as communities of consumers of the research that is produced, examined, and then comes out of systematic reviews. So a little more about some of the core attributes of SRDR Plus, which is the current version of SRDR. It's a collaborative workspace. We can do a set of tasks that are common in systematic reviews: screening, data extraction, data and report sharing, and modularization. As you probably are familiar with, a lot of systematic review reports are huge. So they need, what this tool helps you do is, at the end of the project, modularize the report a little more. It supports multiple users, it's web-based, it's flexible, and it sits on a relational databases. The various aspects of a form for a given study are connected with each other through tabs. And as I mentioned, it facilitates updating evidence synthesis activities such as systematic reviews. But I want to reiterate the key principles of this platform is that it's free, it's open source, and once the data are made publicly available, or once the project is completed and the authors choose to publish the data, it's free and open access to the data. All right, so I'll spend a few slides now walking you through some screenshots of the platform. For those who've used the platform, this should be very familiar to you. For those who've done systematic reviews but might not have used the platform, the structure of the platform should be, and the layout of the sorts of things that you can do on the platform should also be familiar to you. But for those who are not familiar to systematic reviews, I'll walk through a set of different aspects of what we collect in systematic reviews. And the reason I'm showing you some of this is to inform some of the next pieces that I will be talking about, of what comes out of the system, and then hopefully where Maria Michaels will take that from how do you think of that from a guidelines perspective. So I'm sort of setting the stage by doing this. So this is what the screenshot of the, I guess, the homepage of the website is. Once you're in as a user, you have multiple projects that you potentially can be working on, and this is a dashboard that gives you information about those multiple projects. So for example, this is one project that we are currently working on. It tells you how many members are there, how many key questions or research questions there are, how many citations. These are citations that made it to the full text extraction phase, how many different extractions are related to those citations, and SR360 is something that I'll come back to later on. But this is sort of a dashboard that shows you all your different projects, and you can click on any one of these to expand on them. Within a given project, you have a whole list of study extractions that you can, especially me as a lead of this particular project, I can look at how many studies are in there, who are the users on this project who extracted data for each study. I can reassign using functionality here and progress on each of the studies. And so this is a list of study extractions. Once you're in a given extraction, by an extraction you mean for a given study, the team has extracted data for that study, and this is how it's laid out within the platform. It's laid out into a set of tabs that are organized in the left here. You can see key questions, designs, arms, outcomes, and so on. And within each of these, there is a set of questions that I, as a lead of the project or anyone else who's leading the project, can set up questions on the data extraction form that your team members can answer. And these sort of features are reasonably common across the systematic review platforms. There's a set of different types of questions that are allowed. You're not seeing on here sets of matrix type questions as well, but this is a bit of a breeze of a demo that I wanted to go through. But there are multiple choice questions here. There's open-ended questions that you can allow your extractors to answer and so on. Risk of bias. Dr. Alpa talked a little bit about different risk of bias systems that are out there. What we've done in SRDR, we've pre-populated the system with some of the most common risk of bias systems that are out there. And so one doesn't have to look up the tool, type the questions in when you're designing a form. So a lot of these are pre-populated, but not all of them. So some of them will need to be typed in. So these are risk of bias tabs. Questions are here, for example, did the authors use an appropriate analysis method? One can answer yes, no, probably yes, probably no, or no information. So this is from the Robin's Eye tool. For those who have done systematic reviews, you're probably familiar with that tool. And then you can add information or comments related to a particular assessment. Study arms. So how are the treatment arms in a given study? So this particular study had, I guess, 4 arms that were being compared, or treatment groups that were being compared. You lay them out on this tab. And then you have a tab that's known as sample characteristics, which is you're describing the group of participants in each of those study arms at quote unquote baseline. So you have, again, those 1, 2, 3, 4 arms that I talked about, and a total for across all arms. You have a set of questions then related to age. You can ask, there's further down, there are questions related to sex, race, body weight, body mass index, or you can have weight as well, and any other variable that you want to collect about participants at baseline. So this is the sample characteristics tab, and this is the outcomes tab. I'm going to spend a little more time on this tab than I did on the previous tabs, because it's going to inform one of the things that outcome definition view, or that example I wanted to talk to you about. So the way this is laid out is you have various types of outcomes in the review. For example, mortality, length of ICU or NICU stay, length of hospitalization in overall, I guess, not just in ICU. So you have 3 outcomes in this study for this review. You have information about when the outcomes were measured, and then you have a set of things that I'll come back to you. So for now, just skip over the red circle. I'll come back to the slide to talk about what you're looking at here. But there's information about, you can add a new outcome, that you can talk about various populations that for whom results were reported for this particular outcome in this particular study. And you can add subgroups if necessary, and time points as well. So this is the outcomes tab. For each of those outcomes, I mentioned it's a relational database. So when you get to the results tab, it's already recognized that you've added this outcome to this review. You have multiple arms in this particular example that we're looking at. You can tell the extractors can extract information such as what factors were adjusted for. These are new, and then you have a set of numerical fields related to how many events occurred in that particular arm in this particular study. What was the percentage of participants? What was the baseline, and so on. So you have a set of things here to get at that information. And then you have, so those are arm specific pieces of information, and then you have between arm comparisons. For example, you have an odds ratio with a 95% confidence interval and a p-value for specific comparisons between arms in the study. So this is the results tab, and of course where you can click on this and get to another result and enter similar information. The 2 quadrants in the below that don't have data here, you can also, for continuous measures especially, you can have changes within arms from baseline to a time point, and you can also have net comparisons, sort of difference of differences in that cases, or net mean differences. So this is how the results tab is laid out. One of the functionalities that SRDR Plus also offers, and this is also for the purpose of this talk, it's a bit of an FYI, it allows the data extractors or a third person to compare extractions between 2 copies of a given article, just to make sure that there were no differences between the 2, and then if there were differences, to resolve them. So it's sort of data comparison tool if duplicate extraction is done in the systematic review. All right, so that was about using the platform as to help do the systematic review, but what about accessing data from systematic review that already exists? So one of the things SRDR does, I mentioned the open access policy related to once data are published. So you can access published systematic review data from this website at the bottom, the published projects data. You click on these, you can download Excel files and other formats for, depending on what was submitted and what was uploaded through the process of SRDR as end users, so as guideline developers, or anyone in the community. We wrote a paper here. I just put the DOI on there, and you have access to these slides after the fact. If you wanted to look at a description of these various published projects, we wrote this paper last year where we summarized what's in there. There are about 150 at the time, but now we are up to about, I think, 180 or 190 published systematic review projects with available data. All right, now let me come back to a slide Dr. Alper showed related to this ecosystem. So he talked about production of evidence and synthesizing of evidence and the next step of using that evidence to inform guidance and so on. So this corner or the top left of this circle is where SRDR Plus sits, using that synthesized evidence, putting it together in a systematic review. What I want to show you now is a little bit of an example of one piece of how we are trying to think about this, the next sort of step in the circle, in this ecosystem, if you would. What comes out of the systematic review and making that a little more computable and interoperable. An example of this is the outcome definition viewer that our team has worked on. Let me first tell you what it is, and then there's important credit that I want to give as well. So what it does, it incorporates the FHIR standard that Dr. Alper so nicely explained, using the EBM on FHIR Resources to make the systematic review data that are in the platform and so variable, but to make it more computable so that it can be easily understood by the next system, or easily accessed and then turned into some sort of a human accessible way by the next piece in the system, or the next player in the evidence ecosystem. It's just an example of that. The outcome definition viewer is one simple example of that. It's not everything, it's just the outcome. But before I show it to you, I want to give complete credit to our lead programmer here at Brown University, who's been working on it, Jens Yap, and our assistant programmer, Birol Centurk, and of course, Dr. Alper and others as well, in that evidence evaluation and tools development work, reporting tools development work group that he mentioned. So they've been the architects of this work. So I come back to that view of the outcome viewer. So what I'm going to show you now, for each particular outcome in their systematic review, we've developed ways in which to express information about the outcome, not the results yet. We've just focused on the outcome. So everything that I'm going to show you is information in each row of this table. You have information about that outcome in a JSON format, in a computer readable format, and then in a more human-friendly version, which is the outcome definition viewer, or ODV. So I'll start with the JSON format. I won't spend too much time on this, only to show you that this is the resource that this is described, and it's the evidence variable. And it's laid out in computer readable format, but it has pieces of information about the description of the outcome, as I'm going to use that example of that first one there, mortality at 30 days. So you can see mortality type not specified. You can have types such as all cause, cancer specific, and so on. So in this case, it's type not specified at 30 days. And you have the SNOMED code for it that's listed here. The display is dead would be the finding, which is number of patients who died. And then the time points is at the bottom. So this is sort of the JSON definition of 30-day mortality from this in FHIR format. A more human-friendly version is what we have been working with, as I mentioned, with Computable Publishing and other collaborators to develop. And that's this outcome definition viewer. So what you're looking at here is a URL. You can click on this and access that JSON code that I just showed you. So that's the source code. The human readable description of this is in that description field that says mortality, type not specified, blah, 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 30 days. How components are combined. This particular outcome has just one component, mortality, but for those familiar with composite outcomes, such as mortality or myocardial infarction, in such cases, each of those components would be described here. But in this case, there's just one component because we're just talking about mortality, and then coded content related to the code for for dead being the outcome of interest there. So that's the code from the SNOMED code, and then the coded content for what the value was in terms of the time point. 30 days is 30 days, is 30 is the quantity, and day is the unit. So you can see how where we started with was here. You have a computer readable code and then a more human friendly version of that same particular outcome. And this is my last slide, or my last substantive slide, I guess, which is here are my my closing thoughts, which is evidence synthesis is highly resource intensive. We need efforts like this to promote interoperability so that we can be talking to other systems, reduce redundancy. Many efforts are underway. Dr. Alpo went through a lot of those. I'm not going to repeat those. And hopefully tools such as SRDR+, as I mentioned, it's just one of the tools that are out there. The outcome definition viewer example that I showed and other ongoing efforts can serve as examples for other demonstrations of data sharing so that the next step of the process, which is the guideline development process, which many in this audience are particularly keen on, or working on, or interested in, and the rest of the evidence system and those steps are facilitated. So that's our where I think this sort of example of a use of our system we think could be potentially helpful. With that, I'd like to wrap up and say thanks to GIN North America for inviting me, AHRQ, of course, for funding SRDR, and all of our users and stakeholders in the evidence synthesis community, and of course to all of you for your attention right now. Thank you. And I will hand it over to, there you go. I'll hand it over to Maria.
Okay. Well, hello, everyone. Let me know if you are able to see my screen.
Yes.
Okay. And you can see my title slide.
Yes. All
right. Excellent. I'm sorry I'm having some technical issues, so I'm glad that you guys can hear and see me. I'm going to start with a couple of sort of refresher slides. So Brian started out talking about the healthcare ecosystem as it is today, where it's not quite forming a well-run cycle, and the different parts of our knowledge ecosystem don't quite fit together as best as possible. We want to then move to this desired healthcare knowledge ecosystem where all the parts and pieces kind of move together smoothly in this cycle and sort of help move the other one along. You heard both Brian talk about the data to evidence part of the cycle in particular, and Ian talk about a tool in SRDR Plus that can help make the data to evidence part of the cycle computable. And I'm going to focus a little bit more on the evidence to guidance part of the cycle. And after me, you're going to hear from Ed, who's going to talk about tools in CDS Connect that can help with this part of the cycle. So just to start with, let's think about what it would take in order to review all the volumes of information in the published literature. And you can see here from a study back in 2004 that this is about 627.5 hours per month, which if you look at it kind of in a pie chart, assuming about an average of 720 hours a month, that's a pretty big chunk of your month that you're reading this published literature. And then this tiny sliver to sleep, eat, see your family and friends, see patients, complete clinical documentation, et cetera, et cetera. If you break that down into number of hours per day, we're talking about 21 hours of your day reviewing literature and a little over 3 hours doing everything else in your life. Basically, this is not feasible. So in addition to the time aspect, it's actually also not very feasible for the human brain to retain all of that kind of information, especially not to the degree of detail that's needed. So this is when clinical decision support as a tool started to be more and more used. This was several decades ago. It was meant to help in clinical decision making, really for all parts of clinical care. The problem is that this did not come without issues. And over the years and decades, really, these issues have been tried to be worked on, but we haven't completely succeeded in making clinical decision support work effortlessly. So at the CDC, we decided to bring together a lot of different stakeholder groups involved in guideline development and implementation into something we called a Kaizen event. Kaizen is a Japanese word that means continual improvement, and it is actually part of the lean methodology. If you've heard of Lean Six Sigma, it's the lean part of that. And we called this meeting Adapting Clinical Guidelines for the Digital Age. So this was about 2.5 years ago. The reason we went this route is because this method incorporates all the relevant perspectives, both strategically and tactically from the beginning. It can achieve big changes in short order, and it's very transparent among the participants, which contributes to a high level of buy-in, and it really helps each of those participants really better understand the challenges from each perspective. And so I'm not going to read this slide to you, but hopefully you can tell by looking at this list of stakeholder groups that we really did try to think of all the different types of groups that are critical to guideline development and implementation and brought them together. And in some cases, just as an example, some of these groups hadn't talked to each other, such as the guideline authors and all of the informatics folks that worked on the health IT standards and developing things like clinical decision support and clinical quality measures. So just by having those interactions, we started to better understand each other's perspectives, challenges, et cetera. So at the end of the day, what we identified was that some of the big problems in guideline development and implementation include long lag time, inconsistencies and inaccuracies in translation, and all of this helps contribute to this average of 17 years for scientific evidence to apply in patient care. And a big reason is that we're essentially playing the telephone game with the guidelines. So we have these multiple translations of the guidelines, which add complexity, opportunity for error, and variation across different healthcare organizations or sites, and sometimes even providers within the same site. So those of you from healthcare organizations probably know that quite well. And so what we determined was that a solution to these problems could be developing the tools and the guidelines together so that we can help the evidence apply to patient care more easily, quickly, accurately, and consistently. So now going back to today's guideline development and implementation process, again, having this pretty long implementation time, and this goes back to how we do this. We start out with looking through research results, doing literature reviews and meta-analyses, and after that review, pulling together a group that then determines what evidence makes sense to update guidelines or produce new guidelines. Then there's a guideline narrative that's produced, and even if it is, quote, digital, it's really still in just PDF format. Really, it's just a narrative. And so that gets released, and each and every organization that should follow that guideline ends up looking at the information and going through a fairly detailed process, of which I'm representing a pretty high level version of that process here. And this pretty complex process to interpret the guidelines happens for each and every organization, for each and every guideline, or really each and every recommendation. So if we take that just within the US and look at organizations that use certified health IT, now we're talking about being this whole process being performed by up to 96% of 5,500 hospitals and up to 86% of 355,000 clinics, which means that we have a lot of unnecessary redundancy in the way that we interpret and implement guidelines. So one way to think about the way that we translate the evidence into something that can be used in practice, used within a workflow, such as in executable clinical decision support, is this idea of different knowledge levels. And these knowledge levels aren't really sequential or anything. They're just really different. You think about them as different formats of the same information. So in level 1, we have the narrative format. And an example of this would be a guideline that's for a specific disease that's written in the format of a peer-reviewed journal article. I'm sure everybody on this call is very familiar with that particular knowledge level. Then we start to get more and more structured. And we moved to L2, which is semi-structured. And now we're talking about things like flow diagrams, decision trees, and other similar formats that describe the recommendations in a slightly more structured way, and they are human readable. Then we get even more structured with L3. Now we're talking about a standards compliant specification that encodes logic, uses data models and terminology and code sets, incorporates value sets. It's basically ready to be implemented, and it is computer readable, but it is not yet implemented. And that brings me to the final level of knowledge, L4, which is executable. And an example of this would be clinical decision support that is implemented and used in a local execution environment, such as a local EHR production system. Could also be incorporated through web services. There's something called a Smart on FHIR app, which is kind of like the apps on your phone, where they sort of sit on top of the platform that is your phone. In this case, the analogy, the phone is the EHR. So there's different ways that this can be executed within the clinical workflow. So the other part to this is the process of actually getting from guidelines to that implemented, executed clinical decision support. So as we thought through this as a multi-stakeholder group, it really seemed like we had to redesign guideline development and implementation. So now going back to what it currently is in a slightly different view, we've got the guidelines that are translated to things like clinical decision support, which then have to be incorporated into clinical workflows for patient care. And if you think about the different perspectives that are needed to do this, you've got your guideline authors that produce the guideline in the first place. You've got communicators that help disseminate and kind of spread the word that a guideline update or new guideline is available. You've got the informatics folks that translate that into different kinds of informatics tools that can help with the implementation. You have those implementers at the local healthcare organizations that take those kinds of tools and incorporate them into clinical workflows where they ultimately reach patient care. And maybe, but not always, there's some sort of evaluation that happens. And I say maybe, but not always, because it truly is inconsistent. And while we have things like clinical quality measures that are a form of evaluation, most of the time those are treated somewhat separately and really thought about as regulatory requirements more so than quality improvement tools. So what we really end up having is this inconsistent or even non-existent feedback loop back to the guideline authors. So in the way that we've redesigned the process, it's a lot more parallel and iterative, where we have all of those different perspectives represented at the outset, and we have concurrent guideline development and translation, where we can iteratively work with the implementers early on, and again, iteratively, so that we can take into consideration not only the science, but also the implementation considerations. And we can also plan in advance for some of that evaluation that doesn't currently happen consistently. And if we do all of that, when we're ready to release the guidelines, now we have more of a package of the guidelines and other kinds of tools like clinical decision support and clinical quality measures that can go along with the guidelines that have been tested pretty broadly, so that we can get that scientific evidence into patient care as quickly as possible. And then if we execute on the evaluation that we planned for, we can consistently close those feedback loops. So in order to do this, and Brian mentioned FHIR earlier, you also heard Ian talk about how that gets incorporated into tools like SRDR Plus. We developed this implementation guide called FHIR Clinical Guidelines, or Clinical Practice Guidelines, CPG on FHIR. And again, this is using FHIR, which is an interoperability standard that's intended to facilitate the exchange of healthcare information between organizations. What we did was we took that standard and we sort of specified it for use in clinical, making clinical guidance computable. And that's what's in this implementation guide. I've got the links down below, so if you wanted to take a look at it, you could. The current draft is in the first link, and we expect it to be published before the end of the year. So I already have the URL there for when that happens.
Maria, 1 minute.
On FHIR. Was somebody speaking? I'm sorry.
Just time check, 2 minutes.
Okay. So the CPG on FHIR is an international standard through a standards development organization called HL7. It includes this standardized and scalable approach that can help translate and implement the clinical practice guidelines and other types of guidance more efficiently and effectively. It provides a framework for improving the knowledge ecosystem and includes some related common health IT standards. So the key aspects include that integrated process I described, those common standards that can work really across that entire life cycle that we talked about, the knowledge ecosystem, as well as different EHR platforms. And then it can provide this closed loop between the guideline content and information flow, so we have these feedback and feed forward processes. So how does this work in practice? Well, the computable guidelines, again, these levels of knowledge are progressively more structured. It starts out with this narrative, which we're all familiar with. It's basically a prose recommendation, for example, and then it becomes more and more structured. So we have the semi-structured L2 version, like flowcharts, definitions, functional descriptions. Then you become even more structured through adding the terminology and data elements and logic, and it becomes this computable recommendation. And both of these levels you can actually see in the link at the bottom there in the blue strip, which is in CDS Connect, which Ed is about to present to you. And both of those together can be used to then ultimately execute this information, the evidence in the recommendation, into the clinical workflow in the EHR, and that would be the L4 level. So what this boils down to is the computable guideline that is really this one translation that can be implemented in many different ways. So if you think about this as kind of a stack of Lego blocks, you can reconfigure the Lego blocks in a multitude of different ways in order to implement it. So you can use the same computable guideline, for example, to incorporate clinical decision support within the workflow, clinical quality measures that can help you track how well you're doing or how well the outcomes are, as well as for other forms of data collection, for example, electronic case reporting. And what this really comes down to is we can use standards like FHIR to transform that health data landscape from our current sort of multi-approach methods to a more streamlined future state where we have more of a common method that streamlines our ability to exchange data in a way that actually gets that evidence into practice, into patient care a lot more easily, quickly, accurately, and consistently. And if we do that, our wheels will smoothly spin and we'll be able to get that desired healthcare knowledge ecosystem. So with that, I will leave it here and turn it over to Ed for more information on CDS Connect.
Okay, so Maria, maybe you can make me presenter. I can't. There you go. Thank you. Great. Okay. So hi everyone. My name is Ed Lomaton. I'm the last in the series of presentations, and I'll try to go relatively quickly. I know there's some good questions hanging out there in the chat and in the dashboard. So as mentioned at the top, I'm Ed Lomaton. I work at the Agency for Healthcare Research and Quality. And similar to Ian, I was given the task of talking about a platform called CDS Connect, Clinical Decision Support Connect, which we've developed at AHRQ through our engagement of the Health Federally Funded Research and Development Center. This is an FRDC at the department operated by MITRE. Before I dive right into CDS Connect, I wanted to set the context a little bit and give you some background. As probably many of you know, AHRQ is a probably best known as a health services research agency. We fund health services research, including clinical informatics topics such as decision support. We've been funding decision support topics or grants and contracts actually for as long as I can remember. In fact, my very first presentation at a research meeting was on behalf of the Glides project, which many of you may be familiar with. This is Rick Schiffman's project from now 10 years ago. But this effort that we've taken on at AHRQ more recently really began in 2016 with funding from the Patient-Centered Outcomes Research Trust Fund, the PCOR Trust Fund, which was set up by the Affordable Care Act, and then that fund was renewed last year. And it really has two broad aims, initiative, to one, advance evidence into practice through decision support, and to try to make CDS more shareable, interoperable, and publicly available. I should mention that we interpret or think of decision support in pretty broad terms for the purposes of this program, meaning, and I continue to find the 5 rights framework probably the most easily understandable. It's the 5 rights most usually attributed to Jerry Ostroff and others when we describe CDS as bringing the right information to the right people using the right channels and formats, meaning the right technologies at the right times during workflow. To try to do that, we've set up over the last several years basically 4 pillars or components. One is to engage a stakeholder community. That's basically you. So me doing this presentation is part of that arm or set of activities. We engage patients and clinicians, informaticists, guideline developers, and many others. The second piece is really to try to create some prototype infrastructure for sharing and developing interoperable CDS. This is basically CDS Connect that I'll dive into a little bit more. The third aspect is recognizing that we continue to recognize that CDS is a ripe area for research, and these funding opportunities are really around how do you demonstrate and how do you conduct research that can demonstrate how CDS can be used to advance evidence into practice. How do you put PCOR findings into practice through decision support? We know there are lots of interesting questions that you're answering out there, and those funding opportunities are for that purpose. And we have an overall evaluation component. So you might be wondering, well, what exactly is CDS Connect? It is many things at once. Perhaps first and foremost, it's a place. So you can go to the web. It's a website, a web page. You can go dial in cds.arc.gov. That's the landing page for our overall CDS initiative, but you can easily navigate to CDS Connect from there. And you'll find essentially a set of things that you can do and discover. One of which is probably the heart of it is a platform or repository. There you can find what we call entries in or artifacts. These are entries like on a library of sorts where people have contributed interoperable CDS resources, puzzle pieces, if you like, those bits and pieces that help construct clinical decision support in a more shareable, interoperable way. I'll come back to this. We have a set of tools that we've developed, and one of which is probably best known is the CDS authoring tool. It helps people write decision support rules that come from guideline recommendation statements into an interoperable standards-based computer interpretable language. And maybe most important is we have a community that supports and drives the project overall. Maria is actually has been our co-chair since the beginning. And I'd encourage you to please contact me afterwards. There's actually a contact us functionality on the CDS Connect site where you can join the work group. It is a public work group. We have about 130, 140 different organizations represented, maybe 80 or so more unique individuals. We meet monthly. It would be great to have you participate. One of the things that I think the strongest theme that I think comes through most through our presentations today is sort of, you've seen many graphics around circles and cycles and feeding off and learning from each other. This is no different in terms of CDS Connect. As Maria talked about, the process of translating guideline recommendation statements into decision support that people can reuse depends on people reusing and learning from each other. That is really at the heart of why does CDS Connect exists. So that the repository, you'll find those entries I talked about, and mostly that level 2 and level 3 representation that Maria talked about. The structured and semi-structured forms, the if-then representations, and also the computerized computer interpretable forms. Our project has used or adopted use cases as a way of demonstrating the infrastructure, both the tools and the repository. We've adopted use cases around cholesterol management, around chronic pain, around preventive care. We also very much depend on FHIR as a way of anchoring our standards work or interoperability work. You can see the types of FHIR standards that we leverage within the project. And essentially in this quote unquote life cycle, the idea is that people are posting and reusing those artifacts, those puzzle pieces, that code, those level 2 and level 3 representations, iterating and improving upon them, reposting them, and the cycle goes around again so that we can increase that efficiency as Maria talked about. I mentioned tools. So the CDS authoring tool, as I said, is anchored to Health L7 clinical quality language. It's that HL7 standard that defines the logic in a rule that might come from the guideline recommendation statement. We have other tools that are available, such as the CDS CQL testing framework that allows people who are generating those rules and algorithms to test them. And we have tools that are around figuring out how to integrate that algorithm, that CQL, into the workflow through the EHR through something, for example, called CDS hooks and LHL7 standard. These are all open source. They're all freely available. And you're seeing right here a screenshot from the landing page of that CDS authoring tool. Imagine, a future state where you're writing guideline recommendation statements and the CQL at the same time. Wouldn't that be great? And wouldn't that sort of reduce the level of interpretation, perhaps around going from the narrative to the computer interpretable guideline form? To be a little bit more concrete about that, I'm just going to take you through the chronic pain management use case or example. You don't have to read everything on this slide. I think you'll get the gist of it. On the left, you see the pros. This is very familiar to all of us, how most guidelines are written and presented. It's very efficient. It's really effective. Most clinicians absorb and digest guideline recommendation statements in this form. As you go to the semi-structured or level 2 representation, you have to convert it to if-then statements, again, something that Maria talked about. And the other thing, I think, reason for putting this up here is, as Maria talked about, there is a need for greater specificity, greater precision to enable computer interpretation. That's not in the original guideline, whether that was intentional vagueness or not. And the process of making these jumps really needs to be exposed so that the next sort of, say, user or reuser of that artifact can know what the assumptions were, what interpretations were made, so that they can decide for themselves whether they would make the same interpretations. Same thing goes for the translation to the next level, the level 3 CQL version. Lots of interpretations and assumptions and choices are made around what value sets to use, what terminologies to use. How am I going to represent this data element in my computer system? That stuff needs to be exposed if you're going to trust and be able to share all of what's around that decision support or algorithm for reuse. Lastly, there's that executable form, the user interface that gets executed on a local level. What you're seeing is basically a screenshot from the pain management summary, which is a Smart on FHIR app. Maria mentioned that as well. This is an open source Smart on FHIR app that tries to bring together all the different pieces of a patient's pain history into one view in the electronic health record, which is often found in disparate places in the EHR through most user interfaces. We are, the other sort of aspect around pain management in this use case, and sort of brings home the point of CDS Connect, is we continue to iterate even within the projects. We have separate contracts around pilot testing and building upon the pain management summary, building on the CDS so that we're testing it in live operational clinical settings. And in this case, actually adding patient views and patient ability to enter or add to that data that is shown to the dashboard. Also, ongoing work around integrating this dashboard around prescription drug monitoring programs. This graphic comes back to something that the patient-centered CDS Learning Network, another funded activity from AHRQ. It really, again, goes to this notion of cyclic or learning nature of everything that we do. Decision support, like everything else that you've seen, that those gears that we've seen a couple of times, has its own set of cycles where, again, we need to learn from each other and not reuse those things where we don't have to reuse, putting patients and families at the center, recognizing there are a lot of external factors as well that impact our work. A couple more things, and then I'll bring it to a close. One of the things that I think comes through the themes for today is around automated or fair access to evidence and the building blocks of guidelines. The CEDAR project is a new initiative at AHRQ. It stands for CEPI Evidence Discovery and Retrieval. CEPI is itself an acronym. That's the center that I work in at AHRQ. And this is the programmatic home for the evidence-based practice centers, systematic reviews that Ian talked about. It's also the home for where our support for the U.S. Preventive Services Task Force, and you'll find those recommendations, also the home for CDS Connect. The idea behind CEDAR is to create and prototype a standards-based API so that folks who want to access those evidence repositories through other platforms, apps, for example, can access it through that API. so that innovators, other folks can develop those apps, develop those platforms to let clinicians and others get access to it without having to have a human browse real time, say at the point of care or other times, looking for information. I have a link there. Please come ask me about questions. We're just starting that off in the last couple of months, but it's super exciting, as you can imagine. And lastly, I think it's worth telling you and reminding you again, the need for research to demonstrate how to use CDS to implement evidence into practice, say through guidelines. This is an active R18 funding opportunity for grants that's out there now. This first received in January. Please do take a look at that FOA if any of this is interesting to you. And its purpose, again, is to try to demonstrate how to get evidence into practice through scalable, interoperable decision support. Coming back as a bookend to the very first slide, I think Ryan showed earlier, the ACTS slide. Again, lots of cycles and circles. We have taken, I think, a whirlwind journey around the circle, maybe more than once. And I encourage you all to look at the ACTS homepage, where a lot of this is happening. And I know that I saw a lot of folks on the webinar today who are participating in that ACTS work. So I'm going to stop there, and hopefully we have some time for questions. That's my contact address, or maybe I'll leave it here. Thank you.
Hello everyone. Can you see my screen? Hello everyone. Thank you for joining us. We have come to the end of our presentations from our panelists today. We encourage you to, we have a few minutes for questions, actually about 7 minutes exactly. We encourage you to send general questions and suggestions and comments regarding this webinar to the GIN North America general email box. We hope to see you for more webinars in 2021, and also look for our announcement for the GIN North America March 2021 two-day virtual conference and open space meeting. Before we try to answer some of your questions live, I want to turn your attention to the question and answer section. We had suggested or requested questions to be submitted in the chat feature, but we noticed that questions were being submitted in the questions section. And several or all of our panelists have been trying to answer your questions, so we are suggesting that if we do not get to all your questions today, that you switch over to the question and answer section to see if your question was being responded to. One of the first questions we received was for Dr. Alper. If you would like to speak to whether or not the computable tools for quality improvements, or is it limited to research? I believe you responded to that question in the question section, but in case you want to verbalize your response.
Well, there were 3 questions, and they may have been asking the same thing in different ways. And some related to quality improvement, some were just asking, you know, is the use of these tools available for free? So I'm not sure if they were separate questions or not. The tool that I demonstrated, Clinical Trial Results Reporter, was a tool to express a certain type of data, you know, the statistical findings related to evidence variables, and have that transformed into FHIR Resources, computable expression of that evidence. That could be applied to a quality improvement, you know, measure. If that's the type of data, you know, what is the outcome findings, what are the variables? It could be applied to systematic review results or observational study results. The particular configuration that I showed was clinical trial results, targeting that type of approach, but we will develop that tool further to other types of data expression, and it's a tool to convert, you know, human data entry for evidence and statistics, and then produce the FHIR-enabled tools. The tool suite to do that will take software development, and we will have to pay engineers to develop it. So I cannot say that all of the tools will be open for free for all things. We have developed an accelerator model to make it much lower cost than traditional subscription to support the development of the tools. And we also have committed that anything related to COVID knowledge with these tools will continue to remain free through the COVID Knowledge Accelerator. And I'm happy to work with anybody individually to, let's say, be generous in specific, circumstances. But I do have to find the sustainability model to support the ongoing scaling and sustainable development of these, of these tools. That may even be a conversation with the GIN as an organization, at some point. But so I would encourage you to not be deterred by that. And if you want to use these tools early while we're still setting those up, just let me know, and we could figure out how to set you up with it. At the moment, it's available free on the web, but we will have to develop more structures and login systems and other things with these tools. Will make them as accessible, as widely accessible as possible, because that's the intention of these tools development, is to support this broad infrastructure.
And one other question that came in is that what are the anticipated additional resource requirements for de novo digital guideline development, and what are the possible pathways for digitizing existing guidelines? Can anyone speak to that?
Sure, I actually just answered that in the question box, but essentially we don't have the specifics yet. That is actually something we need to work on figuring out the more we do some piloting and actually start to use these different standards and the integrated process that we described and all of that in practice. But kind of generally, it does involve having, in say a guideline development group, informatics expertise, implementation expertise, and working together in parallel in an iterative way back and forth, so that you can actually have potentially a better narrative. You have a faithful representation of the narrative in a computable version, and then you've tested that computable version at the local level, at least with a representative sample of local organizations, to be able to determine if there are any local implementation challenges that need to be taken into account in the more general computable representation. So there would be a more, I'll just say, top-heavy resource requirement, but if we do this right, it should then alleviate all the unnecessary redundancy downstream, where every single healthcare organization is essentially doing the same thing over and over again.
Two quick responses to that question. I'll take it in two parts. What are the anticipated additional resource requirements for de novo digital guideline development? And the ACTS project, I believe, is a space where across the broad community, answering that question, saying these are the requirements, spelling out that vision from the broadest set of perspectives, so that we have a global view to what are the requirements, I believe is a space where there will be active work to answer that question. And then the second part, what are the possible pathways for digitizing existing guidelines? I've shown you some tools, and that was for human data entry form. We've worked with some others. How can we convert from existing data sets, if it's structured data, to those forms? And if you have digital guidelines already in a digital form that's structured, we may be able to work with you. And at this point, because all the structures are non-standard, it'll take individual work with each group how to convert that to a standard. So that's something we could follow up with people individually, how to convert from what they have now, to a standard. And over time, as tools develop, that will support shifting common forms, to a standard.
Okay, thank you. And just a reminder, everyone, as we mentioned earlier, the slides and a recording of the webinar will be available in about a week or so. You will need to have your GIN account information to log in and access the slide deck and the recording of the webinar. So if you desire to review the information discussed here today, you may choose to do so by downloading the webinar through the GIN website. Also, the panelists, their email information, contact information is available on their slides. So upon reviewing their slides, if you have further questions, you may feel free to contact them. Thank you so much for joining us today. We greatly appreciate your time and see you all in 2021.